Good morning, students. Today we will see another topic in Unit 3, that is PDH complex. PDH means pyruvate dehydrogenase complex. So this PDH complex is an example for multi-enzyme complex. Before going into this PDH complex, we have to know some introduction. So coming to the metabolic fate of pyruvate. So glucose on glycolysis, it produces pyruvate. So, under aerobic conditions, pyruvate is converted to acetyl-CoA, which enters the TCA cycle to be oxidized to carbon dioxide. Directly, pyruvate cannot be entered into TCA cycle. So, for the entry of pyruvate into TCA cycle, it has to be converted into acetyl-CoA. And this conversion of pyruvate to acetyl-CoA is done by pyruvate dehydrogenase complex. And during this conversion, ATP is generated. Generally, glycolysis is taking place in cytoplasm. So, pyruvate is generated in cytoplasm. But TCA cycle takes place in mitochondria. The pyruvate which is produced in the cytoplasm cannot enter into the mitochondria. For the entry of the pyruvate into mitochondria, it has to be converted into acetyl-CoA, and then this acetyl-CoA will enter TCA cycle. So, the pyruvate is transported into mitochondria by the help of a transporter called pyruvate transporter. Now, we will discuss about the pyruvate dehydrogenase complex. So, this pyruvate dehydrogenase complex is inside the mitochondria, and this enzyme oxidatively decarboxylates the pyruvate to acetyl-CoA. So, pyruvate is oxidatively decarboxylated to acetyl-CoA by pyruvate dehydrogenase complex. This pyruvate dehydrogenase complex is a multi-enzyme complex which contains 5 co-enzymes and 3 apo-enzymes. So, pyruvate dehydrogenase is an example for multi-enzyme complex containing 5 co-enzymes and 3 apo-enzymes. Now, we will see the types of co-enzymes present in pyruvate dehydrogenase complex. The first one is thiamine pyrophosphate TPP, Co-enzyme A, FAD, NAD+, and the fourth one is lipoamide. So, lipoamide is nothing but the lipoic acid, which is also called as thioctic acid, which has 2 sulphur atoms and 8 carbon atoms in it. Next, it can accept, this lipoamide will accept or donate hydrogen atoms. So, this is about the co-enzymes present in PDH complex. Now, I said that PDH is a multi-enzyme complex. That means it is containing multiple enzymes in it. So, what are the multiple enzymes present in the PDH complex means? First one is pyruvate dehydrogenase, which is represented as E1. And the second one is Dihydro Lipoyl Dehydrogenase, that is enzyme 2. And the third enzyme is Dihydro Lipoyl Dehydrogenase. Dihydro Lipoyl Dehydrogenase, enzyme 3. That means pyruvate dehydrogenase complex is containing 3 enzymes. So it is called as multi-enzyme complex. Now we will see the functions of enzyme 1, that is pyruvate dehydrogenase. So this enzyme, it will decarboxylate. It catalyzes oxidative decarboxylation of pyruvate. And for the action of this enzyme, it requires TPP as cofactor. And coming to TPP is nothing but thiamine cofactor, thiamine B1. So, it is a B complex group vitamin mainly essential for the utilization of pyruvate. So, for the utilization of pyruvate, thiamine is required. The 2 carbon unit remains attached to the thiamine, and the pyruvate will be converted into hydroxyethyl-TPP. Then, this hydroxyethyl group is oxidized to form an acetyl group and then transferred from TPP to lipoamide, which is the second coenzyme, to form acetyl lipoamide. Okay, now, next coming to the role of enzyme 3, which is the last step in oxidation So, at the end of the reaction, the cofactor namely TPP, lipoamide and FAD are regenerated. The FADH2 transfers the reducing equivalents to NAD plus to give NADH and H plus. And then this NADH will transfer or it passes to the electron transport chain to give or to generate 3 ATPs. So from one pyruvate, 3 ATPs are generated. In case of glucose, 2 ATP will be produced, 2 pyruvate will be generated by oxidation. So as a result, we will get 6 ATP. So 6 ATP from 2 moles of pyruvate formed from glucose by oxidative phosphorylation. So this is the slide which represents the mechanism of pyruvate dehydrogenase. So the pyruvate will undergo oxidative decarboxylation and the side chain is added to thiamine pyrophosphate. As a result, hydroxyethyl pyrophosphate is produced. And here, this reaction will take place in presence of enzyme pyruvate dehydrogenase. Then, the hydroxyethyl group is transferred to the lipoamide, which is the second coenzyme. In presence of the enzyme Dihydro Lipoyl Dehydrogenase. As a result, the lipoic acid will be reduced, and the side chain is transferred to the acetyl CoA, as it is acyl CoA, coenzyme A, and as a result, acetyl CoA will be produced, and the reduced lipoic acid will be regenerated. That means it will be oxidized by transferring the protons to the FAD. As a result, FAD is reduced to FADH2, and this FADH2 is again regenerated by transferring the reducing equivalents to NAD plus, such that NAD plus is reduced to NADH plus H plus, and these reducing equivalents will move to the oxidative phosphorylation, and for every one NADH, 3 ATP will be generated. In case of glucose, 2 pyruvates will be formed. As a result, 6 ATP will be produced. So this is the energetics for conversion of pyruvate to acetate CoA. Now we will see the regulation of pyruvate dehydrogenase complex. So pyruvate dehydrogenase is a good example for end product inhibition. End product inhibition, that is acetyl CoA and NADH, which are the end products of the reaction, will control the PDH. And next, PDH is also regulated by phosphorylation and dephosphorylation reactions. Generally, PDH is active when it is in the dephosphorylated form, and it is inactive as a phosphoenzyme, which is a reverse case. Generally, enzymes will be active in the phosphorylated form, but pyruvate dehydrogenase will be active in dephosphorylated form. And next, the PDH phosphatase activity is promoted by calcium and magnesium in presence of insulin. So this is about the regulation. So this slide represents the process of regulation of PDH. So PDH complex B, which is the phosphorylated form. I said that phosphorylated form is the inactive form. So this PDH phosphorylated form is represented as B. In presence of PDH phosphatase enzyme and insulin, the phosphate group will be removed. That means dephosphorylation will occur and it will be converted into the active PDH complex, active dephosphoenzyme. And this again in presence of ATP and PDH kinase. Kinase is the enzyme which is adding phosphate group to the enzyme. It will be converted into inactive form. So this is the regular cyclic pathway, and this is the regulation pattern of PDH. Coming to the significance, the calcium released during muscle contraction stimulates PDH for energy production. Next, PDH kinase is promoted by ATP, NADH and acetyl CoA, while it is inhibited by NAD plus CoA and pyruvate. Next, in presence of high energy signals like ATP and NADH, the PDH is turned off. That means in presence of end product, that is ATP and NADH, the PDH is turned off. Now we will see the other properties, that is lack of TPP, that means thiamine pyrophosphate, the coenzyme or the cofactor which is required for second enzyme, inhibits PDH activity and causes accumulation of pyruvate. So once the pyruvate is accumulated, what will happen? It shows feedback inhibition. In thiamine deficient alcoholics, pyruvate is rapidly converted to lactate, resulting in lactic intolerance or lactic acidosis. So, in thiamine deficient alcoholics, pyruvate is converted to lactate, which results in lactic acidosis. In patients with inherited deficiency of PDH, that means in patients with hereditary lack of PDH, results in lactic acidosis. So this is about pyruvate dehydrogenase composition and function, significance and the type of reactions carried. Regulation. So thank you all.